Okay, you have a guided note packet to follow along with on this lecture video. And the purpose of giving you that was that all of the diagrams are on there, so hopefully it'll just be really easy for you to write next to each diagram the different things that are going on, because there are a lot of diagrams involved in this. I also want you to pay attention to the far left-hand side of the packet, left-hand side on this side here, because this is going to be where, as you're watching the video, if you're thinking to yourself, what is she talking about, or I'm just not understanding this, that's a place to write your questions so that when you come back to class, I can go through your questions quickly and answer them, and then we can move on and get to the cat. This lecture video is going to look at skeletal muscle composition, so how is a skeletal muscle organized, all the way down to like the organelle level, and the sliding filament theory, which is basically all of the series of events that has to occur in order for your muscles to contract. So those are kind of the two things that we're going to look at: what a muscle is made out of and what has to happen in order to make that muscle contract. All right, some basic vocab. First of all, myofiber refers to a muscle cell. It is the same thing. And a muscle fiber is the same thing as a muscle cell, which is the same thing as a myofiber. So you'll hear those three words used and they all mean the exact same thing. Second is myofilaments. These are going to be two different types of protein fibers that are found inside of a myofiber or a muscle cell, and there are two kinds. There's actin, which is also called the thin filament, and there's myosin, which is also called the thick filament. And in a little while you'll see a diagram of these. These two proteins are going to be what is directly responsible for muscle contraction. Sarcolemma is just the fancy name for the plasma membrane in a muscle cell or a myofiber, and a sarcoplasm is the name of the cytoplasm in a myofiber. So, as we begin our look at the structure of skeletal muscle, there are a couple coverings that you need to know. These are connective tissue coverings. There's three of them. There's the endomysium, the paramysium, and the epimysium. And the job of these connective tissues, they're almost like skin that bundle different parts of the muscle cell. And their job is to just kind of reinforce the muscle and then to help provide the elasticity of the muscle. So the idea that the muscle can kind of stretch and go back to its normal resting length. It also provides an entry and exit for blood vessels and nerves, because your muscles obviously need to have a large amount of blood supplied to them in order to provide them with the oxygen and the other nutrients that they need for energy. And then nerves, because your nerves are going to send the electrical signals that tell the muscles when they need to contract. So first is the epimysium. So if you look at the diagram that I gave you, the epimysium is the very outermost layer of the muscle. So when you cut open your cats and you see the muscles, what you're actually looking at is the epimysium. It's the overcoat of the muscle. It covers the entire muscle itself, and then it bundles structures called fascicles together. And we're going to go from largest bundle to smallest. So we're kind of starting with the biggest structure, and then we're going to go down to the smallest structure. So next are these bundles here of muscle fibers or muscle cells that are bound together by the paramysium. Paramysium would be found right here, and this whole bundle of fibers then are called fascicles. Each one is a fascicle. This is a fascicle, this is a fascicle, this is a fascicle, this is a fascicle. And then each individual muscle fiber then is going to be bound together by another connective tissue skin or wrapping, which is called the endomysium. So you can see here as this one's being pulled out, each of these are individual muscle fibers. Muscle fiber, muscle fiber, muscle fiber, whoops, muscle fiber, and then the wrapping around them. So this right here is going to be the endomysium. So if you think about it in terms of like a full organ, the muscle is the full organ. The fascicles are going to be the tissue, and then the muscle fibers, so this down here, is going to be the actual cell. And then we're going to get to the myofibrils, which are going to be these structures here, which are going to be just like organelles in a normal cell, like mitochondria or endoplasmic reticulum, or any of those organelles. So full organ here, bundles of fascicle makes up the tissue. Each fascicle then is made up of muscle fibers, which are located right in there. And then each muscle fiber, just like because a fiber is the same thing as a cell, each muscle cell is going to have parts inside of them, which are going to be these structures right here. So if we look at each individual muscle cell, then each muscle cell is basically shaped like a cylinder, and these are going to be what's responsible then for actually causing muscle contraction to occur. They're varied length and diameter, so some are thicker than others and some are longer than others. And you can actually visibly see dark bands and then light bands. And these are what the striations are when you looked at these at a skeletal muscle underneath the microscope. Alright, so here are the organelles basically of the muscle fiber. Here is your muscle cell here. Okay, and we pull out a myofibril. That's going to be one of the organelles. And if we section out that organelle and look at it closely, you have this structure here, which shows you the different parts of the myofibril. So you have the thin filament, which is in blue, the thick filament, which is in red. The I bands are going to be what is the light band. These A bands are going to be the dark bands. So again, the alternating I band, A band, I band, A band. This is what gives the muscle cell their striation. There are these lines in the middle of the I band called the Z discs, and what the Z disc does is it holds the actin, which again are those blue lines. And then the I band is going to be actin only. You only see the blue lines here. And the H zone is going to be a section where there's only myosin. If you look really carefully at your diagram, you'll see here that on either side of the H zone, like right here and right here, that has actin and myosin in there. And so again, the relationship between actin and myosin is what is going to cause the muscle to contract, which we'll get to in just a second. So a sarcomere technically is going to be from one Z disc to another Z disc. That is a contractile unit, which we call the sarcomere. So from here, this is just a little bit better picture because it's bigger, but again, you can see the Z disc with the actin attached to it in blue. You can see the H zone, which only has myosin, the red. Myosin is the red or the thicker filaments. A bands, actin only, and a sarcomere is from Z line to Z line. Here's another different way of looking at it. So here, right here is the thick filament, the myosin. And these blue links here are the thin filament, or the actin. Here's a Z disc that's attached to the actin. So again, if you're comparing the muscle to a regular cell, then the muscle is the organ, the fossicle is the tissue, the muscle fiber is the actual cell, the myofibrils, so these structures right here, are the organelles down here. The sarcomere, which is Z line to Z line, is just a portion of the organelle. And then the myofilaments, which are actin and myosin, that would be like the carbohydrates, lipids, proteins that are found in regular cells, like the other types of chemicals that are found within the cell. All right, so that brings us to the sliding filament theory. What goes on between the actin and the myosin that actually causes your muscles to shorten and you can flex and show your muscle? So these are all of the events that result in actin sliding over the myosin and basically causing that sarcomere, so that distance between the two Z lines, to shorten. Actin and myosin don't actually change shape, but what they do is they slide over each other, and that's where the sliding filament, which is the actin and the myosin theory, comes from, is how it ends up being named. So the first thing that has to happen is there has to be an electrical signal that occurs down the length of a neuron. Okay, so remember a neuron was kind of shaped like this. You kind of had the cell body, and you had the dendrites here, and then you had the axon, and then it kind of branched off into the ends here. And so if we were looking at this end up close, this is what it would look like. Okay, so what happens is that electrical signal travels down the neuron, or your nerve. Remember, neuron is just the nerve cell in your body. So it travels down the nerve, and it gets to the very end of the nerve, or the neuron. And when it gets to the end, it actually causes the ends of the neuron to release a chemical called acetylcholine. And this chemical is called a neurotransmitter. So it basically dumps all this acetylcholine, which is the ACH here, into the gap between the nerve, or the neuron, and the muscle. So this is the muscle here, and this is the neuron. So it dumps all that acetylcholine right in between the gap there. And that acetylcholine actually attaches to the muscle fibers on the cell, and it causes calcium to get dumped inside the muscle fiber. So first acetylcholine is released, it binds or connects itself to the muscle, and once it connects itself, all of a sudden calcium just gets dumped into the muscle fiber. That calcium then actually attaches itself to the thin filament, or the actin. So here's the actin here, here's the calcium, So the calcium will just grab onto the actin filament, and it causes little doorways or sites on the actin to open up. So it's almost like once the calcium binds, it's like a key. The calcium unlocks these little doorways on the actin, and when it unlocks the doorway, the myosin, which has this head-like structure here, can actually physically attach itself to the actin filament. So it's going to grab hold of it. Okay, kind of like when you grab hold of the doorknob to open the door. That's what the myosin does. It will attach itself to the actin when the doors are open because the calcium unlocked it. Once that myosin head attaches to the actin, what it's going to do is it's just going to power the actin towards the middle of the sarcomere, and we call this the power stroke. So think about like if you were pulling up a bucket from a well, you grab the row pull and then you grab and you pull. That's what's happening with the sliding muscle theory. The myosin attaches to those openings on the actin, and then it power strokes it forward. So it's going to power stroke that actin towards the middle of the sarcomere. Okay, and when it does this, it actually releases ADP as part of the process here. Okay, so ADP gets released because ATP is being used. Okay, because you need energy to power stroke. So ATP is used and it ends up breaking up into ADP and the inorganic phosphate. Then what happens is other ATP will actually come in and it will attach itself to the myosin head, which will kind of serve like a deadbolt basically. It will cause this to release. It causes the myosin head to release away from the actin, detaching itself. So actin and myosin will always stay stuck together until ATP comes along and allows the myosin to release. The ATP will eventually split apart back into ADP and inorganic phosphate, and so it will release energy, and the myosin head will go back to its original position, and then it'll then wait for the more calcium to become available so that it can attach itself to that open door and then start the process all over again and power stroke and then release. So basically what happens with the sliding filament theory is that you have calcium opens up the doorways on the actin, the myosin head attaches. When it attaches, it will power stroke the actin towards the center of the sarcomere, and then it will release, and then it will attach again and power forward again. Attach, release, attach again, and power forward again. So again, the best example would be like you pulling up a bucket from a well. You attach it with your left hand, you pull. You grab it with your right hand, and your left hand releases, and you pull. And so it's this constant pulling, or in this case, this power stroking to bring the thin filament to overlap thick filament. So altogether, myosin attaches to the actin. Once the calcium joins on, power strokes it forward, releases, gets itself back into normal position to get ready to attach and power stroke again. And it goes through this whole cycle. So the difference between a relaxed and a contracted sarcomere then, if you're looking, here's the I band, or the Z line again. This is a relaxed one, so Z line, this is what we've already looked at. Here's the actin, here's the myosin. You can see all of those heads for the power stroke. If you look at a contracted sarcomere, you can see that the actin has slid towards the middle. It's gone in this direction. This one's gone in this direction. So now you have actin, overlapping myosin. And A band stays the same, but the H zone completely disappears here, like this area where there was only myosin, because now you have actin and myosin, so the H zone is completely gone. And the I band becomes shorter. So that light band actually becomes smaller, but the dark band stays the same. In order for a muscle to relax, that's how a muscle contracts, is going to be once all the calcium is removed, puts all of those active sites on the actin out of reach for the cross bridges, which doesn't allow the myosin heads to attach, which means it can't pull it forward. So calcium plays a huge role in your muscles being able to contract properly, because if there's not enough calcium, then your muscles can't contract, and if there's not enough calcium, then it's going to take it from your bones, and that leads us right back to our discussion in the last unit about the development of osteoporosis. 3 general phases of a contraction: latent period, that's kind of the delay between when the neuron gets the electrical signal and the point in which the muscle becomes contracted. So basically the time when the signal travels down the neuron, releases the acetylcholine, and the calcium binds to the actin. And then you have actual contraction where the thin filaments slide over the thick, and then you have a period of relaxation where the cross bridges can't attach and the muscle returns back to its normal length. The question becomes, how can you produce so much force that, like if you wanted to lift a car versus wanting to lift a pencil, if your muscles are contracting in that way? And the answer to that question is that you have different, what's called motor units. So a motor neuron will basically connect to a certain amount of muscle fibers. Each muscle fiber only has one motor neuron, but a motor neuron can stimulate or dump all that calcium and acetylcholine into several different muscle fibers. The way that you generate different amounts of strength has to do with how many of these motor units, which is the motor neuron plus the fibers that it is in charge of, are activated. So if I'm trying to pick up a car, I'm going to be stimulating a whole bunch of these motor neurons to generate more force. If I'm picking up a feather, I'm going to only be generating maybe one motor unit. Strength is based on how many motor units are being stimulated and how often that motor neuron is receiving the stimulation. That's all I have for this one. If you have any questions, but make sure you're writing them down on the side of that guided note packet so that we can go over those questions quickly and you can get back to studying your cat.